Okay, so imagine you're standing in a library. But this isn't just any library. This one contains the instruction manual for building a human being. You've got about 20,000 chapters, what we call protein coding genes.
Right.
Now, picture most of those chapters locked behind a glass case. You can see them, they hold the keys to things like cancer or Alzheimer's, but you can't touch them. We have drugs keys for maybe, 10% of them?
If that. The rest is what we call the undrugged genome.
The dark matter of biology.
Exactly. We know these proteins exist, we know they're involved in disease, but chemically we just can't seem to get a grip on them.
Because finding a single key for one of those locks can take, decades. But today, we're looking at a paper that proposes a way to unlock all of them. Not in decades, in a single day.
We're talking about a system called DrugCLIP.
And this isn't just finding one new drug.
Oh, no. The mission here was effectively to take every single druggable target in the human genome and screen it against every possible drug molecule. We're talking hundreds of millions of them, all at once.
That just sounds physically impossible. Usually, drug discovery is like looking for a needle in a haystack. This is like looking for needles in 10,000 haystacks at the same time. So before we get to how they did it, we have to talk about the bottleneck. Why haven't we done this before?
Physics. The bottleneck is literally physics. The traditional way we do this on a computer is called virtual screening, using molecular docking.
The old lock and key analogy.
Right. But in docking, the computer has to simulate the actual physical forces. It's trying to push the key into the lock, turn it, check if it bumps into an atom, calculate all the electrostatic forces.
The van der Waals forces, all of that.
Every single groove, it's crunching heavy, heavy math for every single attempt.
And that takes time.
Seconds to minutes for just one pair.
Which really adds up. The paper mentioned that screening 1 billion molecules against just one target, that took 2 winks.
And that was using 10,000 CPU cores. Right. It's just too slow for a genome-wide scale. You need centuries. So the researchers behind DrugCLIP, they realized they had to stop simulating the physics and start treating this like a language problem.
Okay, and this is where contrastive learning comes in. Usually when I hear that term, I'm thinking of, image recognition, teaching a computer the difference between a cat and a dog.
It's the same principle, really.
Yeah.
But here, it's a retrieval task. Think of it more like a translation engine.
A translation engine.
Yeah. You have one language, which is the 3D shape of a protein pocket, and a second language, the chemical structure of a drug.
And DrugCLIP is trying to see if the sentence in the protein language means the same thing as the sentence in the drug language.
Spot on. It encodes the protein pocket into a string of numbers, a vector. Then it encodes the drug into another vector. These two vectors live in a shared space, a latent space.
Mm-hmm.
And the AI's only job is to maximize the similarity, the cosine similarity between pairs that actually work.
So instead of trying to physically jam a key into a lock, it's just checking if their vectors are pointing in the same direction, mathematically.
Yes. And checking the angle between two vectors is a dot product. It's incredibly fast. Millions of comparisons in a split second.
Okay, but here's the catch I always see with deep learning. To train a translator, you need a dictionary. You need millions of examples of this word means that. We don't have millions of solved crystal structures of drugs bound to proteins. We have, tens of thousands?
Even less in the really clean data sets.
Yeah.
And this is where the paper gets, brilliant. They realized they didn't have enough data, so they cheated.
They cheated.
They generated their own training data from thin air, basically, using a method called ProSA.
ProSA.
Protein fragment surrounding alignment.
Okay, break that down for me. How do you get data from nothing?
So the researchers had this aha moment. Proteins are just big folded blobs, right?
Right.
They hold their shape because parts of the protein stick to other parts of the protein.
Like protein hugging itself.
It's a perfect analogy. And that hug uses the exact same physics as a drug binding to a protein. Hydrogen bonds, hydrophobic interactions. It's the same chemistry.
I think I see where this is going.
So they took the entire protein data bank, all the known structures, and just chopped them up.
And if you chop a piece out of the protein, that little piece becomes a fake drug.
And the hole it left behind is a fake pocket.
That is so clever. The protein literally teaches the AI how to bind by showing it how it binds to itself.
And by doing that, they instantly created 5.5 million training pairs.
Wow.
Without a single expensive lab experiment. They taught the AI the grammar of molecular binding before it ever even saw a real drug.
It's like learning the rules of grammar by reading a book before you try to write your own sentence.
Exactly. And once it learned that basic grammar, they fine-tuned it on the real data from the DrugCLIP database to polish its understanding. They even used a tool called RD-Kit to generate random shapes or conformations of the drugs during that training, just to make the model more robust.
Okay, so let's do a quick recap here. The problem. Old school docking is physics-based, it's artisanal, and way too slow.
Right.
The solution? DrugCLIP treats it like a Google search, using vectors and math. But the real innovation, the secret sauce, is this ProfSSA technique, using the protein's own pieces to create millions of training examples out of thin air.
That's the engine. And the speed difference is just, it's laughable. They benchmarked it against GLIDE, which is the industry standard for docking.
On the LIT PCBA benchmark.
Yes, and GLIDE would take about 3 days to screen that data set. DrugCLIP did it in 38 seconds.
38 seconds versus 3 days.
And with modern GPUs, it scales linearly. So you're talking about something that's effectively 10 million times faster.
But speed is one thing. Being wrong really fast doesn't help anyone.
Exactly. You just get a very large pile of useless molecules.
So they had to prove it works in the real world. Which brings us to their first big case study. The NET transporter.
Ah, yes, the norepinephrine transporter, a huge target for things like depression and ADHD. Bupropion, for example, works on this. And for the longest time, we didn't actually have a good structure for human NET. It was only solved very recently, in 2024.
So the AI was kind of flying blind?
Partially. It had to generalize from a relative, a dopamine transporter from a fruit fly, a Drosophila, and these two proteins are less than 60% similar. That's a huge biological gap to cross.
So DrugCLIP goes looking for drugs for NET. Does it just find the same kinds of things we already know about?
No. And this is where it gets a little spooky. See, human chemists have a kind of rule for NET inhibitors. The textbook says you need an aliphatic nitrogen to form a specific salt bridge with a residue called AsP75.
It's the law. If you're designing a NET drug, you put that in.
DrugCLIP broke the law. It started spitting out compounds with aromatic nitrogens.
So nitrogen's locked inside a ring structure?
Exactly. A traditional medicinal chemist might have looked at those and just tossed them. They don't fit the rule.
But they tested them.
They did. They screened 100 compounds and got a 50% hit rate, which is pretty good. But then they went further. They used a cryo-electron microscope to see what was actually happening at the atomic level.
And was the AI right?
It was dead right. The drug wasn't forming that salt bridge everyone thought was essential. Instead, it was forming a T-shaped pi-pi stacking interaction with a totally different residue, Phe72.
Key-shaped pi-pi stacking.
It's just a specific way that aromatic rings can stick together. But the point is, the AI found a backdoor into the protein that humans didn't even know was there. It didn't just copy old drugs, it discovered a completely new chemical mechanism.
That is the dream, isn't it? Not just automation, but genuine discovery. But NET was really just the warm-up act. They went on to tackle something even harder.
Right. Because in the real world, we don't always have those nice, clean crystal structures. More and more, we're starting with predictions from AlphaFold.
And that's the elephant in the room. I mean, AlphaFold changed everything, right? We can predict the structure of pretty much any protein now.
We can, but there's a bit of a dirty little secret.
Oh, I thought AlphaFold solved the protein folding problem.
It solved the backbone problem, the general shape. But for drug discovery, we care about the pocket, the little cave where the drug sits. And AlphaFold is often really messy with the side chains, the atoms that line that cave. They can be collapsed or sticking out at weird angles.
So the pocket is there, but it's cluttered. It's like a deflated balloon.
That is the perfect way to describe it. And if you try to run standard docking on a raw AlphaFold model, it often fails because the lock is jammed. The key can't even get in.
So you need a way to inflate the balloon before you screen it?
Precisely. And for that, they built another tool, GenPack, generation packing. It's another AI, a generative one. It basically hallucinates a fake molecule that fits roughly into that messy AlphaFold pot.
It stuffs a ghost molecule in there?
It stuffs a ghost molecule in, and then it lets the protein physically relax around it. That process forces those messy side chains to move out of the way and hug the ghost molecule. It molds the pocket into a realistic, usable shape that DrugCLIP can read.
It defines the pocket more accurately.
Drastically. And they prove this on what I'd call a boss-level target, TRIP12.
TRIP12 sounds like a sci-fi robot.
It's an E3 ubiquitin ligase. It's been linked to Parkinson's and cancer. The problem? Nobody had ever found a drug for it. 0 known inhibitors. no crystal structure, nothing.
Purely AlphaFold territory.
Purely. So they took the AlphaFold prediction, they used GenPack to clean up the pocket, and then they unleashed DrugCLIP.
Then what happened?
They selected 57 compounds to test in the lab. They got a 17.5% hit rate.
That's amazing for a first pass on a totally dark target.
It's spectacular. They found 2 specific compounds with binding affinities around 11 micromolar. They validated it with multiple assays. It proves you can go from just a computer prediction AlphaFold to a confirmed physical drug laid without ever needing a crystal structure to begin with.
Okay, so let's just recap that whole pipeline because it's pretty incredible. You take AlphaFold for the rough structure.
Yep.
You use GenPack to mold the messy pocket into shape.
Mm-hmm.
And then you use DrugCLIP, trained on those prophase fragments, to screen millions of molecules in seconds.
It's a complete factory.
Yeah.
And that is exactly what they did next. They didn't just stop at TRIP12.
They ran the entire pipeline, the genome-wide part of the title.
They took 10,000 human proteins, which covers something like 20,000 different binding pockets.
Yeah.
And they screened them against 500 million molecules.
The scale is just, it's hard to wrap your head around.
It comes out to 10 trillion scoring operations. 10 trillion.
How long did that take?
They did it in under 24 hours, using only 8 A100 GPUs.
If you tried that with the old docking methods, you'd be waiting until the next ice age.
Easily. And the result of that 24-hour sprint is something called GenomeScreenDB. It's a completely open access database. It has 2 million potential drug hits covering nearly half the human genome.
But we have databases like Chembly, right? How is this different?
Chembl is a record of the past. It shows what we've already found for about 4,800 targets. GenomeScreenDB is a map to the future. It covers nearly 10,000 targets, including all the orphans, the proteins nobody has studied because they were considered too hard.
And they pulled out some specific examples in the paper that just show the potential here.
Oh yeah, like SLC45A2. It's a transporter protein involved in melanoma. Historically, very hard to drug. DrugCLI found molecules that seem to bind right near a critical stability site.
And what about Oro6A2?
That one is fascinating. It's an olfactory receptor. It helps you smell. But it turns out it's also active in immune cells and plays a role in atherosclerosis plaque in your arteries. And DrugCLIP found potential inhibitors for it.
So we might be able to treat heart disease by targeting a smell receptor.
That's the kind of wild connection you can only find when you screen everything against everything.
It really feels like we're moving from an artisanal era of drug discovery, you know, one chemist, one lock, one key,
to an industrial era. It's becoming a search engine query. This combination of AlphaFold for the structure, GenPack for the refinement, and DrugCLIP for the screening. That's the new holy trinity.
But I have to play devil's advocate here.
Go for it.
So we have the map, we have this incredible vehicle, we have 2 million predicted hits for half the human genome generated in 24 hours. Why don't we have the pills yet?
That's the million-dollar question. The bottleneck has shifted. It used to be finding the molecule. Now the bottleneck is the wet lab.
You can't swallow a hard drive.
You can't swallow a hard drive. We can predict 2 million hits, but we can't synthesize and test 2 million compounds in a day or a month or even a year.
Yeah.
We have to validate them. And then you have clinical trials. The computer can tell you a molecule binds, but it can't tell you if it's toxic to the liver.
So the computational chemists have thrown the ball over the fence, and now the experimentalists have to go find it.
In a way. But it also changes the job of the medicinal chemist. They aren't just designing molecules from a blank slate anymore. They're becoming more like curators or prompt engineers for these huge data sets. They look at the top 100 hits from DrugCLIP and use their human intuition to say, okay, this one looks promising, this one looks toxic, let's test these 10.
It empowers them instead of replacing them.
Exactly. They start on the 90-yard line instead of at their own goal line.
Before we go, here's a thought I've been chewing on. We've talked entirely about proteins. But proteins are just the machinery. The actual instruction manual is written in RNA.
You're thinking, why wait for the protein to even be built?
Exactly. If DrugCLIP can learn the language of protein pockets, could it learn the language of RNA folding? Can we drug the instruction manual itself?
That is the next frontier. It's incredibly difficult. RNA is so flexible, so wiggly, it doesn't have stable pockets like proteins do. But if we could crack that code, we wouldn't just be treating diseases. We'd be editing the outcome before it even happens.
The tools are out there. The database is public. If you're a listener, a researcher, a student, GenomeScreenDB is open. You could, in theory, look up a protein for a rare disease and find a predicted drug for it right now.
That's the beauty of open science. The barrier to entry for making a massive discovery has never been lower.
Stay curious, check out the data, and keep questioning the bottlenecks. Thanks for listening to The Deep Dive.
See you next time.